Recording in progress.
Okay. All right. So, hi, I'm Dr. Thomas. Welcome to the Biological and Biomedical Sciences Seminar Series. This is the last of our semester, so we're really excited to have Dr. Jason Heindel here. He earned his Bachelor's of Arts at Amherst College and his PhD from Harvard, and then he did a doctoral fellow at Indiana University. He arrived here last year, so he's one of our newer faculty, and we're really excited to have him here to present integrating cell cycle regulation, persistent infection in Agrobacterium.
All right, thank you very much, and thanks for the opportunity to present to the to the whole department. And if you haven't seen my talk before, I hope you learn something from it and enjoy the talk. And if you've seen it before, it's like getting to watch a favorite movie again. So I hope you enjoy that. There will be some fluorescent images here and there throughout the talk. So if you cannot see something, tell me and we can maybe temporarily lower the lights so you can see what you want to see, and then we can raise them up again for those taking notes. And if you can't hear me, of course, raise your hand or something, flail at me. And as I always tell my students, I usually give them a time in the classes that I teach, a time by which I want to stop talking and make sure you have an opportunity to talk at me or with me. 1 hour from now, 3 o'clock, if I'm not done, flail around and let me know that I need to be done. Okay, just like cut me off. I should be done before then, so we have plenty of time for a discussion or a conversation about whatever. So we'll get back to what this image is as we come. This is like a teaser for what's happening with our system. Let's see if I got this right. No, I don't. I need to click on this. There we go. Down. Okay. All right. So I'm going to talk to you about crown gall disease. This is a disease, a plant disease that you may not be terribly familiar with it, but this is what it looks like. This is a gall at the crown, a crown gall at the interface of where a particular plant species would be buried in the soil. So the soil would be down here maybe, and the gall would be right here forming on the tissue of the plant. This is plant cancer for all intents and purposes, although it's strictly tumor formation. It's not a metastasis. So it's just overgrowth, neoplastic growth of plant tissue that is generated by infection from a number of organisms, in particular the organism we study in my lab, Agrobacterium tumefaciens. So it's ubiquitous, it's all over the world. There are all different varieties of crown gall disease and all different pathogens related to Agrobacterium that cause things like grapevine gall disease, also caused by an Agrobacterium species, or hairy root disease, also caused by a particular species of Agrobacterium. So a very common organism, well known, doesn't typically kill plants, just reduces their economic productivity, and we are not excited about that if we're trying to produce a crop or trying to increase yield of our crop. What's interesting about it is the mechanism by which this gall is formed has been used by scientists now for decades for genetic engineering of plants. And I'll show you in 1 minute what that means by genetic engineering of plants. So Agro is a member of the alpha proteobacteria class of bacteria. It's a gram-negative bacterium. There are a number of commensal and pathogenic alpha proteobacteria with which we come in contact all the time. Agrobacterium is one of them. There are plant symbionts that are mutualistic symbionts. They provide a resource to the plant. Those are nitrogen-fixing organisms like Sinorhizobium or Rhizobium meliloti. They provide nitrogen to the plants and ultimately provide nitrogen to you and me when we eat the plants. And then there are some animal pathogens as well, like Brucella, which causes brucellosis, and that causes brucellosis in cattle and sheep. And if you are dealing with those trades or eat some raw milk cheese, you can get brucellosis. So it will infect you and cause things like endocarditis. As one example of an alpha proteobacterium. Other alphas that you may be familiar with are the rickettsial diseases, like Rickettsia rickettsii, so Rocky Mountain spotted fever. So very common, very well-known group of bacteria, very important. And the pathways and the things that I'm going to talk about in this talk are conserved in these species. So even though we work with agro, we can translate our findings into these other systems potentially. So agro is itself and has been for decades a well-studied system, a well-established system for studying type 4 secretion. I'll talk a little bit about what that does and one of the things it does. Quorum sensing, which is a real fun thing that bacteria do and other organisms do, and for the ecology and evolution of facultative pathogens. And the facultative aspect of this will come up in a few minutes as well. It's increasingly being utilized as a good system, agro is, for studying type 6 secretion. So that tells you there's at least 6 types of secretion. And for studying something called cyclic di-GMP, which is a second messenger in many bacteria. And in agro, it's a very useful second messenger that we study, and for biofilm formation. So if you are interested in bacterial biofilms, agro is a pretty good model for studying certain aspects of biofilm formation by bacteria. Here we go. Here is a cartoon. It's a wonderful diagram from 2019 depicting the transformation process. And by transformation, what we're talking about is taking genes from the bacterium and transferring them into the genome of a plant. And this is what is normally and naturally done by Agrobacterium to cause disease. And what it does is, here's our bacterium. So it's a gram-negative bacterium. So they've got 2 membranes. Here's one outer membrane, which you can kind of see. Then there's an inner membrane in the periplasm space, which is kind of, it doesn't normally ruffle off like that. That's just for, so you can see what's happening. In the cytoplasm, you have some of the genomic material. Not all of the genome is present or shown in this diagram, but Agrobacterium has 2 chromosomes, and our species of Agrobacterium has 2 megaplasmids. So unlike what you might read in the textbook for microbiology, where you're taught that E. coli has 1 circular chromosome, and that's what bacteria have, that is true for E. coli. It has 1 circular chromosome. Other bacteria have a different array of genetic complements or replicons that form their genome. Agro is no different. It has those different components. So what happens is, when the bacterium is interacting with its host, which could be one of a number of plants, typically dicots, but there are dozens and dozens of plants with which it might normally interact. This interaction between the bacterium and the plant cell, there's communication of signals that are secreted by the plant or that are produced by the plant because of normal metabolism of the plant in the rhizosphere that results in a change in pH in the rhizosphere near the plant, a change in phosphate concentrations near the plant surface, and this is in the soil, and also secretion of certain phenolic compounds and something called acetylsyringone, which is a molecule generated by the plant, secreted into the roots, into the rhizosphere area where the bacterium may encounter it. These signals all ultimately result in activation of transcription of a set of genes that form a type 4 secretion system. That type 4 secretion system is encoded on a plasmid called the TI, or the virulence plasmid. And this plasmid contains all the genes necessary for genetic engineering of the host plant. And what it does, this type 4 secretion system builds this machine that can then translocate proteins and DNA across both membranes of the bacterium, and then also across the membrane of the plant host cell. So the bacterium itself generates a molecular machine that can then take components from inside the cytoplasm of the bacterium, push them all the way into the cytoplasm of the host cell. And what agro does, what it normally translocates, is something called the tDNA, or the transferred DNA. And so you'll see here this tDNA. It's a single-stranded piece of DNA. When it gets translocated, it's coated with bacterial proteins. So agro is translocating proteins that it has generated and a piece of DNA it has generated. It's coating that DNA with agro proteins into the cytoplasm. That DNA then traffics into the nucleus of the plant cell. Once it's into the nucleus, it then becomes integrated into the genome of the plant. And once it's integrated into the chromosome, one chromosome somewhere in the plant genome, this is a permanent transformation of that plant. So that plant has now been genetically modified. It's been engineered with whatever genes were on this DNA, this T or transferred DNA, they have now become a part of this plant cell's permanent genetic information. And if that's an embryonic tissue, then this plant could then divide and become a completely genetically engineered plant. So that would be a stable transformation. Normally for agro, the genes that are transformed code for 2 things. They code for the biochemical capacity to make something called openes. Openes are a food source for the bacterium. That's one thing that's coded on the tDNA normally. The second thing that's normally on the tDNA are analogs of plant hormones, these phytohormones that are made by, encoded by the bacterial DNA, transferred into the plant nucleus. The plant then expresses those genes. Those hormones result in proliferation of the tissue. So Agrobacterium is transforming plant tissue so that it makes food for the bug and then replicates more tissue that makes more food for the bacterium. So this is a very clever way that the bacterium is able to use host processes to establish its own niche that it can really, really go down in. Okay, so I've only shown one bacterium here. There's usually more than one bacterium during this interaction. So this is just for illustrative purposes. Oh, there we go. All right, so while bacterium uses this plant transformation to establish a food source for itself, we can actually take advantage of the plant transformation because in that transferred DNA, that tDNA, we can put whatever genes we want, pretty much. So if we want to put something like green fluorescent protein into the tDNA and push it into the plant using the Agrobacterium, we can do that. And then that plant cell will then express green fluorescent protein and glow in the dark. It'll be a nice fluorescent cell. So we can use it for doing all sorts of genetic engineering of plants. We determine what tissue do we want to target, what phenotype we want to affect in the bacterium. And it's used all the time. Here in this table, I'm highlighting just barley as one example, but it's been adapted for use to specifically engineer many food crops and other plant crops that may not be for consumption, but for producing some resource that we might want to do. In this table, what they're showing, this is from a 2006 paper, they're showing in barley, they can target a whole number of tissues of the barley plant itself. And they're showing that you can include a number of different promoters or reporters. These are all eukaryotic promoters driving gene expression of the transgene that's been introduced, or viruses of eukaryotes. So here's the actin promoter, here's a cauliflower mosaic virus promoter, and you can add whatever gene, a couple of other genes to ferry them into the plant tissue for whatever purpose you want. So this is just standard plant biotechnology. What I want to highlight here, though, is this transformation efficiency. It's super low. So this ability that this bacterium does, even in nature, it's super low. This is 4% efficiency of trying to transform plant tissue using Agrobacterium. And we would like to, we scientists would like to increase that number. So that's a big outstanding question, is how can we make this process more efficient? I'm just highlighting down here, if you're reading down here, these are some of the different kinds of phenotypes that have been affected in barley, for example, improving the malt production in barley, resistance to barley diseases, so things we might want to do with particular plants. So it's been adapted to many food crops. There we go. Transformation can be transient or stable if it gets integrated into the genome of the organism in an embryonic tissue, and then you produce a whole transgenic plant. Awesome, that would be a stable transformation. In some cases, you don't get integration into the genome, but you get replication, like plasmid-borne replication inside the plant cell, and that would be a transient transformation that can go away over time. Transformation efficiencies are low, and they vary widely. That's what I was trying to highlight here. And site-specific integration of transgenes, where it lands in the genome, is all over the place. We're starting to develop tools where we can specifically integrate in the plant genome where we want to put our particular gene of interest, but that's in the early stages. So it's pretty much, until recently, it was pretty random. Finally, actually maybe not finally, we can also deliver CRISPR systems using Agrobacterium. And the advantage of using CRISPR systems, one, is that you're not making a transgenic plant. So you have concerns about genetically modified organisms going into your food supply with transgenes from another organism. CRISPR delivery is a way of getting around that, because instead of introducing a gene that becomes a permanent part of the particular plant, you deliver the CRISPR, you can delete part of the plant genome, you can rearrange the plant genome, but you're not adding a new gene to it. So that's something that's new, being recently utilized. There we go. And then currently, we actually use plant-produced recombinant proteins, where these plants were produced by agrotransformation, this process, to make a number of vaccine components, food additives, a lot of manufacturing processes in food biotechnology use agrotransformation to do this. So it's an important process that you're affected by, even if you don't realize it. It's happening in your food supply and in your life. Okay. I like Agrobacterium as well because I believe that manipulating agro and its ability to efficiently transform different plants can help us potentially, I wouldn't say achieve, that's a strong word, but approach better completion of these things called the Sustainable Development Goals. So the Sustainable Development Goals are a set of goals, 17 goals established by the United Nations, and a number of parties to the United Nations have signed on to this, that by 2030, we are going to satisfy all of these 17 items. They're pretty lofty. I agree with you. But I think that using agro transformation can help us address a number of these, including zero hunger, because we can increase food supply by making less organism susceptible, disease susceptible plants, good health and well-being, climate action. We can increase CO2 assimilation, life on land in general by helping plants live better lives. So that's some ways that agro can be used to achieve these sustainable development goals. I call this translational pathology. Others may use that phrase, translational phytopathology, to mean something different. So for translational phytopathology, I'm just meaning that understanding molecular mechanisms, because we're going to look at molecular mechanisms in my research that I'll show you in a minute, governing bacterium-host interactions will allow us to better control those interactions. And that we can control those interactions in two ways. One, perhaps we can prevent the bacterium from interacting with the plant, so prevent gall formation, prevent that disease. Or two, maybe we can increase interactions in a productive manner for increasing efficiency of transformations that we want to happen. So a couple of ways of thinking about affecting these interactions of agro, the bacterium, with its host to maybe achieve these goals or to work towards achieving the sustainable development goals. All right, so back to our gall. So I've given you some background and hopefully some ideas of what motivates the work that we're doing. And so how are we going to go from those lofty ideas of looking at transformation and we want to increase plant transformation, but we're a microbiology, bacteriology lab primarily, bacterial genetics primarily. How are we going to use what we do in our lab to study mechanisms that can contribute to an improved stable site-specific transformation of plant tissue? And so, maybe. Question's harder, that usually works. There we go. Ah, there we go. I don't know why. That was cool the way it went black. I guess if you, okay. All right, so here's how we're going to do it, okay? If you walk into my office, some of you have seen this if you've been in my office. This is my whiteboard. And here I've got the two primary regulatory pathways that we study in the lab. A number of you in here actually are currently working on the VTLR-ABCR pathway right there. A number are working on this pathway called the PdhS-DivK-CtrA pathway. It's a lot of letters I'm just throwing out at you. Don't worry about memorizing them. But these pathways we have been working on in the lab for years. And they were originally identified and studied mostly because we were interested in biofilm formation by Agrobacterium and related to bioformation with motility. However, recently more data have been increasingly provided to say that actually plant transformation is controlled or is partially regulated by these pathways. So now we're like, ooh, we're really excited. These pathways might actually help us address this stable genetic transformation of plants. So I'll just do that. I'll do that again. One more time. There we go. There we go. So just these two pathways, these are the pathways studied in the lab. I'm only going to focus on one of these today primarily, but you can ask about the other if you'd like. So when we think about Agrobacterium in the rhizosphere, and so when I say rhizosphere, I mean the soil near surrounding plant roots. You can think of a couple centimeters away from the root. Typically, Agrobacterium, and I said that we'd come back to this, is a facultative pathogen. That means it doesn't require a host to survive. It can survive just fine out in the soil with no plant host around. But if there is a suitable plant host around and it can sense those chemical signals, it will chemotax towards the root or towards the plant surface or towards a wound of the plant to eventually associate itself in what I call a stable association, which I would eventually call persistent colonization. So stable association with the plant allows for biofilm formation. Once you have biofilm formation happening, you can have quorum sensing happen, and I'm going to get to this in a minute. And once quorum sensing is happening, you can get plant transformation happening. Now, in Agrobacterium, quorum sensing is used for getting the TI plasmid and conjugating the TI plasmid to other members of the community. It's not used for establishing a biofilm, but it is instead activated within a biofilm for conjugation of this particular plasmid. So we kind of look at the order of operations there as inverted from what we often see in the textbooks. All right, so the process for this is you have a motile bacterium in the soil, and it translocates itself to the surface of a plant cell. Once it's interacting with the plant surface, there are two things that can happen. One is pretty much nothing. It can just irreversibly, it can reversibly associate. It may not decide to anthropomorphize it to attach to the plant surface. The second alternative is that it can reverse, this is the reversible, sorry. Then it could potentially irreversibly attach. Irreversible attachment is dependent upon formation of this thing, this orange thing called the unipolar polysaccharide. This is a polysaccharide, a sugary substance that is generated at one pole of the bacterium, which is why it's called the unipolar polysaccharide. The unipolar polysaccharide is absolutely required for attachment to surfaces and for biofilm formation. If you get rid of the UPP, you do not form a biofilm, and you reduce all the phenotypes that might happen during a biofilm formation. So once UPP is deployed, and I will say that the UPP is actually deployed in a surface contact dependent manner. So we can watch under a microscope bacteria hit a surface and then deploy the UPP. And there have been studies not done by our group, by collaborators, where they can actually determine what are the surface properties that most efficiently deploy the UPP, all sorts of interesting things going on with that. But it is typically surface contact dependent deployment of this adhesion. Once it's formed this stable association, the bacterium can then form a microcolony as it divides and more bacteria associate with the surface there. Ultimately, you can get biofilm formation, which is elaboration of the biofilm matrix. In the biofilm matrix are all sorts of goodies. Not only is the UPP present, but cellulose is actually secreted by the bacterium. So cellulose is another polysaccharide generated by agro that forms a major component of the matrix, or the extracellular polymeric substances forming the matrix of the agrobiofilm. There are other things in there that we don't know. Presumably there's extracellular DNA there. We haven't established that that's true, but presumably there's extracellular RNA that's been deployed there for one or another reasons. We haven't established that, but presumably it's there. It's there in other biofilms. And presumably there are proteins that are there doing some activity, maybe maturing the biofilm. We haven't established that in our system. So the matrix is full of items that allow for stable association with the surface. And then at some point, we know that the bacterium can disperse from the biofilm, from the plant surface, and go and colonize a new niche as needed. If the conditions have changed, you have no more nutrients, then the bacterium can sense, it's time to get out of here. And so some bacteria will leave the biofilm as they bud off from the surface. All right, there we go. Let me make sure that was right. Yes. All right, so a couple of years ago, well, actually 5 years ago now, whoops. So maybe more than a couple of years ago, 5 years ago now, we're in COVID time here. So 2 years ago, but it's okay, 5 years ago, this paper came out, and this is like the dream. At the time that this came out, this was like the dream experiment that I wanted to do. This is when I was doing my postdoc. Now I did not do this experiment, but boy, I wish I did, because this is an awesome experiment. So what they did is they actually compared a RNA-seq data set, so looking at all of the genes transcribed in a bacterial population not attached to a plant, and those attached to the plant. So differential expression, what genes are differentially expressed when they're interacting with the plant host. Beautiful experiment, relatively straightforward, maybe. Regardless, they did it. They got some great data. And so here are all the data. Lots of things are either upregulated or downregulated. Things that I'm interested in particular is that upregulated components are things like plasmid conjugation. So we have two plasmids. This is the virulence plasmid. This is another plasmid called the accessory plasmid. These plasmids, they are presumably activated during association with the host for conjugation into other bacteria. One reason for that is to prevent cheaters. And so if you study the ecology of microbial populations out in the real world, there's often a time when some bacteria will lose those plasmids to get rid of that load, the metabolic load of replicating those plasmids, if the food's provided. But if you force conjugation, and then you get rid of the cheater. So that's one idea as to why they might conjugate once they form attach with the host. So that's kind of cool, right? Another thing that gets upregulated is transformation. So here's the transformation, that type 4 secretion system that is going to transfer this piece of DNA, the tDNA, into the plant cell and eventually into the plant nucleus, where it would become stably transformed plant tissue. Then nutrient transport, and that is all these ABC transporters are upregulated to bring in nutrients that are being secreted into the environment, including the opines that the plant cell is now making for its symbiotic accompaniment. There we go. Downregulated, however, are things like cell division and motility. So once it's stably associated with the plant, it doesn't divide as much. It's kind of like, okay, we're in the biofilm, we're kind of settled, let's chill out for a little bit. And motility is downregulated, which presumably contributes to, if we're in a good place, why are we going to move to another place? I'm going to try to get this to go. There we go. The reason I like this is if I circle these components here, these are all components, or most of these are components of the CtrA-DivK pathway that we already studied in our lab. So that was exciting to see that these were downregulated. Here's CtrA itself, DivK. And I can also say that, there we go. Conjugation of this AP plasmid is under control of the VTLR pathway. So the two pathways that we'd already been studying were now identified in this wonderful study that, hey, they're involved in the plant interface. So now we're starting to shift our focus of our research to not just looking at the nitty-gritty of details of these particular pathways. How do these pathways contribute to plant transformation? So that's one way we can get to working on those goals that we were interested in. Okay, so I want to talk about the cell cycle. This is from the microbiology textbook. If you took micro here or are taking it now, you all purchased and read, I'm sure. Maybe not. So we're actually talking about this tomorrow in class. So the bacterial cell cycle, if you're not familiar with it, you might be familiar with the eukaryotic cell cycle, which I learned as IPMAT. I don't know how you learn it these days, but that's how I learned it. You got, prophase, metaphase, anaphase, telophase, right? Something like that. I don't know if there's any new phases in there, but in my day, those are the phases that were present. The bacterial cell cycle is quite similar. You have to do and achieve the exact same things. You have to duplicate everything inside a cell. You have to then make double the cell size, and then you got to shove everything into each daughter cell, and then you got to split the two cells into two cells. That's it. And so this is the bacterial cell cycle as shown for E. coli, which is a gamma proteobacterium. So here's your starting point with its single circular chromosome. And you imagine over time, the genomic content, the chromosome in this case, duplicates, it starts replicating. The bacterium starts to elongate, and then it's going to cytokinese or divide. Hey, not good with this, obviously. There we go. Oh, and then it divides, a division septum is laid down. You divide into two daughter cells, each of which has all of the genome and presumably all the ribosomes it needs, and any other particular organelles or plasmids that might be around have been segregated faithfully into the two daughter cells. That's the bacterial cell cycle. It's pretty straightforward. We know this often as, okay, so it includes, you can tell I just took this from a lecture. Sorry. So it includes generating new cell material, divided into two cells, genome duplication, all this has to be coordinated. Typically, we think of this as binary fission. So if you've taken microbiology, or even if you haven't, you've learned binary fission, where the cell elongates and everything is separated, and then you split into two. And so binary fission, I'm going to put on this slide another image. Here's another depiction of binary fission. So start at the top. Here's your rod-shaped bacterium. They don't have to be rod-shaped, but it's easier to visualize. And as it grows over time, you can see it's elongating a little bit, and then elongate a little more. And then at some point, it splits into two relatively even-sized daughter cells. This is your classical depiction of binary fission. Notice here that something's labeled bluish, these two poles, one's blue and one's orange. There's a reason for that. And the reason for that is these poles are not the same. They're not the same because one of them is older than the other pole. And this age of a bacterial pole is generated at cytokinesis. And the reason is, once you split this bacterium into two bacteria, these two poles that didn't exist before, they now exist. These are new poles. And by definition, they are older than the other pole at the other end of the bacterium. So you have old poles and new poles that get generated just through binary fission, very common. Now, it turns out that these different poles are actually different and functionally different in many bacteria. I'm going to show you binary fission in E. coli. So here in this image, this is a sped up version of of E. coli dividing. What we've done is we've labeled all of the proteins on the outer surface of the bug. with a red dye, Texas red 6-indimiddle ester. So these TRSE labeled proteins, they're all on the outer surface. And we're just going to watch over time what happens to them. So if I, please work. Oh, there you go. There we go. So if you watch over time, you're watching for just what's happening to the red dye. Whatever is the red dye is the proteins on the outer surface of the bacterial cell. And I think what you can see, one interpretation might be the dye disappears. Maybe it's getting degraded. Something's happening to it. What's really happening is, it's not that it's being degraded, it's just being dispersed so that we cannot see it anymore. It's one, when it's tight together, it's concentrated, we can see it. And then as the bug elongates, that red dye gets dispersed throughout the cell body and we can't pick up the red anymore. But you can see at the very end, there's still some red dye at the end. I think at each pole, at the old poles, there's still dye left. And that is because the old poles of this bacterium, they actually don't turn over as rapidly. There's a lot of junk that gets deposited there. Okay, that's E. coli. So binary fission, elongation, division. Here's Agrobacterium. Same experiment, right? Labeling all the proteins on the outer surface of the bacterium with this red dye. And we're just going to watch over time what happens to all of these proteins. Nope. There we go. So same exact experiment. And I think that you might notice, maybe, that it looks a little different. And so, in fact, you might say it looks a lot different. There are a lot of details that are evident if you study this kind of video enough. I'm only going to highlight the main one, which is this initial cell appears not to have any dispersal of its dye. It's almost like nothing's happening to that cell. And instead of elongating uniformly and dividing in the middle, what's happening is in agro, the bacteria bud from one end of the bacterium. They bud. By definition, then, this end is the old pole. And everything that's happening over here is, that's the new pole of the bacterium. And the cell, the material in the membrane is not being transferred into the daughter cells. So this would be the mother cell and each one's a daughter cell. Cool, so something's different. It's similar to this binary fission, but there's something still a little different to it. So that's agro. There we go. We can actually see some consequences of this kind of pole differentiation and this unusual polar elongation and division in old images. So this is an image of a root hair on peas. And these little guys are the bacteria attached to, this is the root hair. And these little dudes are the bacteria attached. And you'll notice that for the most part, every single bacterium in this image is attached by one pole. They're not lying flat, because I'm going to fall over here. They're not lying flat, they're attached by a pole. That's true. They are attached by one of the poles, because one of the poles is the pole used for attachment. You already know something about that pole. You know that that is the pole where the unipolar polysaccharide is located. And that is true. So here is something else that's really interesting for plant transformation. At the pole that interacts with the plant tissue, where the unipolar polysaccharide is deployed, we also see localization. So this is green fluorescent protein fused to components of the type 4 secretion system that does the transformation of the plant tissue. So not only do we get polar attachment to plant tissue, we get polar localization of the transformation machinery. So that's cool. That tells us that this polar development, whatever's happening at that one pole, is important for plant transformation and interactions. This is just a nice blow up picture of the unipolar polysaccharide. This is green fluorescent protein being secreted, being expressed stably in a soluble fashion inside the cytoplasm of the bacterium. And then this polysaccharide is being labeled with a fluorescent lectin. And it's secreted at one pole and one pole only. If it's not secreted at that pole, or if it's secreted at multiple places, the bacterium has trouble. That's an issue. And here is a nice video that a colleague of mine, Pam Brown, generated, showing interaction of these bacteria with a plant root in live cell video microscopy. So you've got the bacteria attached to the plant root. You can see the plant root there. The green in this case is the UPP, and the red are the proteins on the outer surface of the cell. So everything I showed you in non-physiological situations, or not interacting with the plant host at least, does happen when attached to the host. Polar polysaccharide deployment, that binary, that polar elongation and division, very similar. All right, so here is our depiction of the cell cycle of Agrobacterium and of other alphas. I say in friends of other alpha proteobacteria. They all follow a similar pathway for cell cycle. The bacterium, when it surface, it loses its flagellum, and then it deploys its unipolar polysaccharide. The site at which the unipolar polysaccharide is laid down, we believe, is the old pole. And then you have this other end would be the new pole at this point. Over time, the bacterium elongates from budding from the one end, elongating from just one end. And then just before the bacterium is about to cytokinase, what was the new pole transitions physiologically, biochemically to the old pole. So, and that's because the new poles are going to form right here. So that right at cytokinesis, boom, you're going to have two cells with an old pole, and two new poles generated. This cell is terminally differentiated. When it attaches to a surface, that cell does not leave the surface. It stays, and then other cells can bud off from it, but it does not itself leave the surface. This cell can then perhaps go on and colonize a new niche, or it could just circle back here and become part of this microcolony or this biofilm. And we see both happening. What I want to emphasize down here is that this arrow, the CTRA activity indicator, is something we really are interested in because, hey, get out of here. That the activity of this protein, CTRA, its synthesis, its degradation, and its phosphorylation status, which put those all together, we'll call that CTRA activity. Those are all changing throughout the cell cycle. So it gets more active here, less active, increasing activity, very active, and then you cycle back. So CTRA, it turns out, is a primary regulator of the bacterial cell cycle for almost all of the alpha proteobacteria. In our lab, we study motility and biofilm formation. We use motility and attachment as proxies for activity of CTRA. We're interested in motility. We're interested in biofilm formation. We're really interested in, those of us who work on this project, really interested in cell cycle progression. And so we see these phenotypes, motility, attachment, as readouts, possible readouts of CTRA activity. And so I'll show you a little bit about that. There we go. So I'm going to simplify. This is like the most simple model for the regulatory pathway we study. So if you want to think of what model we're looking at, this is the model to think about. CTRA. It is called CTRA. It is the cell cycle transcriptional regulatory A. That's CTRA. And it is a master regulator, a global regulator of expression of hundreds of genes in Agrobacterium that control dozens of phenotypes, including attachment and biofilm formation, genome replication, motility, and others. The phosphorylated form of this protein controls motility. The dephosphorylated form controls attachment and biofilm formation and genome replication. So its phosphorylation status changes where it binds in the genome to either turn on or turn off different suites of genes. Important for our work is that this pathway is highly conserved among the alphas. So at the top here, these are just different members of the CTRA pathway. So remember I said CTRA pathway can get complicated. If you want to just think about CTRA, that's fine. There's a bunch of other components. And so these are different components of the CTRA pathway. Here's CTRA right here. And this is a phylogenetic tree of a number of members of the alphas. So Agrobacterium is up here at the top. And it has all these components that are listed in this particular layout of the pathway. But you can see that mostly well lose some. So this part of the PdhSsensor is less well conserved. So where there's white boxes, the proteins aren't present, the genes aren't present. But CTRA is pretty much universally, well, almost universally conserved. So well conserved amongst a number of organisms. We do take advantage of that to study other bacterial systems, including Brucella. So this is a schematic of the Brucella CTRA regulatory pathway, where it has some sensor kinases that interact with this thing called DivK, whatever that is, that ultimately result in activity of CTRA that affects growth and replication of Brucella. Brucella is an intracellular pathogen. It actually gets into your cells and replicates in the cytoplasm of your cells. And this activity of being able to replicate or not is controlled by CTRA, so it could be targeted for brucellosis. And so we actually have a couple projects we've done working on using agro to study Brucella's CTRA pathway. CTRA activity has also been shown to be modulated in Sinorhizobium, which is one of these plant symbionts that promotes, it's a mutualistic interaction with plants that fixes nitrogen. And so during bacteroid formation, where plants undergo this sort of terminal when bacteria undergo a terminal differentiation, they kind of stop dividing, they grow into weird wonky shapes, and they fix nitrogen. In fact, that differentiation is controlled by CTRA. And furthermore, that differentiation or that effect on CTRA is controlled by the plants and certain peptides they secrete to control this in plants. So the plant secretes peptides that induces cell cycle arrest in these symbionts in the bacteroid. All right, in the bacteriosome. Here is a pathway that we've developed for the CTRA pathway. This is a model how the CTRA pathway looks in Agrobacterium. It looks pretty complicated. You don't need to know all of it. Remember, you can just focus on CTRA if you want, because that's the main player. It does seem pretty complicated. It turns out it's actually a collection of a couple of regulatory pathways glommed together. So in one case, there's that conserved region, the CTRA pathway, where you have this transcription factor, CTRA, that is regulated via phosphorylation by CCK and ChIP-T. And you have a second group, a second pathway where you have DivK, another regulator that controls activity of, ultimately, this bottom pathway. The activity of DivK is controlled by these particular sensor kinases. So the CTRA pathway is well conserved in alphas, and the PdhS portion is fairly well conserved among some families of alphas, but not in all alphas. There we go. And so they intersect here at the point of DivK and CCKA. These responses, the actual cellular outputs or the phenotypes you might measure, are under control directly of CTRA or of this small molecule called cyclic di-GMP. So cyclic di-GMP, which I mentioned at the very beginning of the talk, it's an intracellular second messenger for signaling within a cell. Okay, now here is sort of the first experimental work that we had started doing in this pathway. And we took the easy way out. We went ahead and made deletions of the non-essential genes. This means all of the genes in that pathway, we could make a deletion of, remove this gene from the genome of Agrobacterium. We did that. And the ones that were essential, we could not do that. So we did not do that. Although we tried because we didn't know they were essential. And this is how we determined that they were essential. Once we made these deletion strains, and so whenever you see the delta, that's deletion. It means we've deleted this gene from the genome of this particular strain of agro. We then did some quick and easy assays that we do as proxies of our CTRA activity. So we do biofilm formation. Our standard biofilm is on a plastic cover slip, set in a well. It's a 12-well plate. So you just set the cover slip in there, incubate it with a culture of your bacterium over some time point, 1 day, 2 days, 3 days. You pull it out, and then you can label the biomass. You can dye the biomass with crystal violet in this case. And so crystal violet is binding to components of the biofilm matrix here. And so this is our wild type, and you can see you get this nice purple biofilm that's forming at the air-liquid interface because the Agrobacterium requires oxygen for growth. And so at that interface is where it's actually forming its biofilm to grow. Correlated with biofilm formation, we measure swimming motility, and swimming motility is done. We typically do them, you can do them in test tubes. We do them in plates. So we take agar that is at a low percentage or low density agar, just 0.25 to 0.3%. Your plates that you're streaking things out on are 1.5% agar usually. So they're a little denser. These are kind of jiggly. And so we inoculate our bacteria into the center of the plate, and then over time we measure simply how far has this group of bacteria moved from the point of origin. And we can see that when we do this with these different mutants, we get different phenotypes. In some of the bacteria, we see decreased biofilm formation, there we go, and decreased motility relative to the wild type. And in some of these bacteria, we see increased purple crystal violet, so increased biofilm formation there. And this one, increased biofilm formation, but not really a huge increase in motility. We can quantify these things, and so they're quantified here. The one that I want to highlight for you is this one, so the delta PdhS2 strain. So if we delete this gene, delta PdhS2, this is one of these sensor kinases that we predict interacts with DivK. If we delete this, we get an increase in biofilm formation and a decrease in swimming motility. And that's kind of nice because that kind of makes sense in that attaching and swimming away from, we think of as opposite actions of the bacterium. So if we affect one, we typically predict, although we can be proven wrong, and so the data will tell us what's happening, but we typically propose that the other will be inversely related to that. So this was nice inverse correlation, up biofilm, down for motility, really nice. So a couple other phenotypes that we can look at just to show you some of the phenotype tests that we can do is we can do stains for components of the biofilm matrix. So here we have Congo red, this is a dye, and it binds to the unipolar polysaccharide and cellulose and maybe some other components in other bacteria, but for agro, these are what we know it binds to. Some of our mutant strains do have increased Congo red, that's the PdhS2 strain. Some of them have decreased, so this DivK strain has decreased Congo red binding. And calcifluor, this binds something called succinoglycan, which is another exopolysaccharide generated by agro. So we told you about UPP, cellulose, now I'm telling you about succinoglycan. There are 6 known polysaccharides made and secreted by agro, so we're not even hitting all of them here. But calcifluor, we know, binds to succinoglycan, and this mutant, for example, binds more succinoglycan than the wild type. Calcifluor is something you might use if you're going to the dentist. They can use it to actually check if you've got a lot of biofilm on your teeth. That's something they can use. You can also use Congo red for that, by the way, just in case you're wondering. If you want to go to the dentist, check yourself before you go to the dentist. So another thing that we like to look at is, and this is like the big money question is, well, we have all these phenotypes that we're interested in, and we're looking at plant transformation. Well, is transformation actually affected? And so what we typically have done in the lab is we transform potatoes. So this is a piece of potato, it's a potato disc, about the size of a dime, if you guys remember or have seen a dime. I don't know if you've all seen a dime these days. It's about this big, it's about this wide, this thick. And so we lay these potato slices onto an agar plate. No nutrition in the media, it's just agar and water. Put the potato tissue down, inoculate bacteria onto the potato. So the only food source is the potato. And then we watch over time, typically 3 to 4 weeks. So it's a long experiment for those of us who aren't working with mammals. So it's kind of a long experiment, but then we start to see these new growths. These are tumors. These are galls, not really galls, but they're tumors. It's neoplastic growth of this plant tissue, which has been produced because of successful transformation of the potato tissue by agro. We can see that some of our mutants, in particular the DivK mutant, it forms fewer tumors. We can quantify that. Fewer tumors, and we already knew that the DivK mutant had different phenotypes. So we're kind of excited about that. Like, hey, this cell cycle regulation appears to be important at some level for the ability to efficiently transform plant tissue. So this is some data that we were excited about. All right, I'm going to pull these up real quick. So here is, I'm highlighting here the PdhS kinases. These are histidine kinases, and they're a set of kinases that you find all over the bacterial kingdom, and even some plants have histidine kinases. And they're called the PdhS kinases based on 2 founding members in an organism called Colobacter, Ple-C-Div-J. So they are the Ple-C-Div-J homolog sensor, PdhS kinases. And so they are typically, they have transmembrane domains. The blue is a transmembrane domain. And then they have a His-Ka domain for receiving a phosphate group and then transferring that phosphate group to a target protein. And then they have an ATPase domain, which is pulling the phosphate off of an ATP, transferring it to a histidine residue on the protein, and so on. And so they're well conserved. What I've highlighted here, these are the protein sequences, the amino acid sequences for Agrobacterium's PdhS kinases and Colobacters. And then this is an outgroup just to show you that these residues that I've highlighted in yellow are not conserved in all histidine sensor kinase. So this is an E. coli histidine kinase. It doesn't have all of the same residues. Important for us, this histidine residue, that is the histidine residue that makes histidine kinases a histidine kinase. It receives a phosphate. This threonine residue we actually know is important for the opposite activity. Instead of for phosphorylating, it's important for dephosphorylating. So many histidine kinases are bifunctional. They can phosphorylate a target protein, and they can promote dephosphorylation of the same target protein. And these 2 residues, the histidine and the threonine, can help differentiate what the protein's doing, what role it's really playing. So we actually wanted to focus in a bit on PdhS2 because it gave us that beautiful inverse phenotype, high biofilm, low motility. So we started looking at it. And just to highlight, the number of PdhS kinase members in alphas varies widely. So Agro has 4 members. Some organisms have 12 PdhS members, which predicted should all interact with the same pathway. So there's multiple regulatory inputs to how to control CTRA activity, which should indicate it's pretty important. There's a lot of ways to control it. There we go. So looking at our pathway, the first thing we wanted to verify is when we would hypothesize in this pathway based on prior work that if PdhS2 is really interacting with DivK, that there would be a genetic interaction between the two. And so these data actually show us that if you get rid of DivK, you do not see your PdhS2 phenotype. So here's when you see down here, the black bars are always going to be our adherent biomass or biofilm. The white bars are always going to be our swim ring diameter, and we're normalizing these to wild type. So our wild type bacterium we're saying is 100%. You get rid of PdhS2, you have that up more biofilm and down less swimming. DivK is decreased in biofilm and slightly decreased in swimming. We can come back to that if you want. That could be a question you might ask. But these are the data. And so we would predict that if there's a true genetic interaction like this, that if we combine these two mutations and make a double mutant, that actually it will phenocopy this strain. And so the data, the data show that. So in fact, the activity of loss of PdhS2 it requires DivK to be present. So that allows us to verify our genetic model that PdhS2 is upstream of DivK and interacting with DivK. So just some of the kinds of the ways we start to piece together this pathway. Next thing we actually did is we wanted to determine, does PdhS2 act as a kinase or does it act as a phosphatase? So if we predict that it's interacting with DivK, we would predict it's phosphorylating it or dephosphorylating it. And to evaluate that, we made different alleles of PdhS2 where we either replaced the histidine with an alanine, so this should not be able to phosphorylate DivK, or where we've replaced the threonine with an alanine residue, and this allele should not be able to dephosphorylate, but can still presumably phosphorylate targets like DivK. So I'm showing you down here again, same idea. Black bars are biofilm, white bars are motility. And as seen before, set to 100% for wild type, delete PdhS2, more biofilm, less swimming. We can complement that mostly with wild type allele of PdhS2 on a plasmid. And so when we do that, we express PdhS2 on a plasmid in this background strain. We reduce biofilm formation closer to wild type, and we bring motility back up closer to wild type. So it's not perfect complementation. If you want to ask about that, we can talk about that. Now what we did is we actually, with what we're calling our kinase null phosphatase positive mutation, this is where we've now moved or replaced this histidine with an alanine. What we can see, so this allele of PdhS2 cannot phosphorylate its target. It can dephosphorylate its target. And what we see is that it actually behaves just like the wild type, pretty much. The data are almost indistinguishable, that the amount of biofilm is similar, the amount of motility is similar. So this gives us the hint that in fact it's phosphatase activity, not phosphorylation activity, that's important for PdhS2. The converse experiment with the other allele, there we go. This shows exactly the opposite. So if we now have a kinase positive, an allele of PdhS2 that can phosphorylate, but phosphatase negative, it cannot dephosphorylate its target proteins, and we express that in this background, it really promotes biofilm formation. Look at this number, 1042, I had to cut it off. And it further exacerbates the motility phenotype of this delta PdhS2 strain. So that kind of tells us that excessive kinase activity is what's driving our phenotypes. We actually interpret this to mean that it's the phosphatase activity that's most important during the normal life cycle of Agrobacterium. So this is a way that we go about looking at how these different proteins do what they do and what their targets might be. A lot of it is genetics and then phenotypes based off of the genetics. So coming back to this, based on our description of binary fission and then our cell cycle of Agrobacterium, and what we now are looking at the CTRA pathway, led us to ask this question here, well, how is CTRA activity actually being affected? And is it different in daughter cells? Or is it, if you have this pre-divisional cell, is there sort of more or less CTRA activity controlling these phenotypes in one cell type or the other? What's going on there? How is that happening? And one way that could happen is bipolar localization of proteins. So what I'm going to show you we've done here is we've taken PdhS2, that protein that I'm super excited about that acts as a phosphatase. We fused it with green fluorescent protein, and then we just watch over time as these cells grow and divide. And so wherever you see a green focus right there, that's where PdhS2 is. And so if we play this, I think you can see that. So if we play this and watch over time, it's sped up, 20 minutes a frame. This is a total of 8 hours of video microscopy. If you watch over time, the cells are going to grow and then they divide. One of them actually bites the dust. It's up here, the one that bites the dust. You'll see it explode. You can watch right now, it's going to explode. Oh, it's gone. Yeah. So, but if you watch the green dot, I think what you can maybe convince yourself, and if you can't convince yourself, what you can do is you can actually trace them. There we go. You can get still images and just yourself trace where the green dot is, where's this cell, what's happening. And you'll see that PdhS2 is a new pole specific kinase. At every cell division, so if you imagine binary fission, the bacterium grows. At every time there's a new cell division, there's a new pole generated, PdhS2 moves to that pole. And then it's at that pole, and then there's a cell division, and it moves to the new pole that was just generated. So PdhS2 is at the new pole. Naturally, this leads us to ask, well, we've got 4 of these PdhS kinases. Where do they all localize? Are they antagonizing one another? Are they at the same location? How is that controlled? So one thing, I'll just show you one set of data for that, maybe. There we go. This is DivK, another one of these kinases, predicted to interact with DivK and ultimately affect CTRA and ultimately affect our phenotypes. And we've fused DivK to green fluorescent protein. Beautiful. And we just watch it over time. Here we go. This one's easier to follow because you don't have to go anywhere. It doesn't do anything. DivK, once it is established at a pole, it is there forever. So DivK is, in fact, an old pole specific kinase. So data like this, we can combine all these data together to build up, to build this model where we have in the standard dividing Agrobacterium with its unipolar polysaccharide at one end, flagella produced for motility at the other end. We have localization at the new pole of PdhS2. At the old pole of DivK, I didn't show you data for PdhS1, but we know that it tends to localize there. Please C has been hard to localize. We and others have not been able to localize it yet. So something funny going on there. We don't know the answer to that. This localization of these proteins and their activity towards DivK should give a gradient of activity where DivK is phosphorylated and active in the older cell, the mother cell. And ultimately the activity of these leads to a gradient of activity of CTRA. CTRA is phosphorylated in the new cell. And so that allows for nice segregation as the cell is growing. And then once cytokinesis happens of the outcomes, where did we go? There we go, of these events. So once it divides, this can stay as a biofilm, this can be a motile cell, and this cell cycle can begin again. So I want to show you what's happening in biofilms for these, because we kind of came back to the idea that, well, we're interested in stable transformation. Stable transformation happens when the bacterium interacts with the plant. Before that happens, it forms a biofilm on the plant surface. And that is true. So, and I know I'm getting close to my time. I'm watching it. So here, this is DivK, which is an old pole specific kinase, right? It localizes to that one pole, the old pole. The old pole is where the bug attaches to things. And so this is a biofilm formed on a cover slip. And now we're doing confocal microscopy of it. We've labeled the membrane of the cells with a membrane dye called FM464. And so you can kind of see the outlines of individual cells. So this is about, I don't know, 3, 4, 5 cells thick. And if you look straight down, you can see a bunch of cells and a bunch of dots. The dots are DivK. If you look along the orthogonal slices through this biofilm, you can see that for the most part, the DivK focus is at the bottom of the surface of that. And that makes sense because the bottom is where things are attached to the surface, which is where the old pole is, which is where the unipolar polysaccharide is. Even more stark is if we make this 3-dimensional image and we peel up the biofilm, Now you can see that, yeah, DivJ is really localized on the bottom surface of the cells, interacting with the surface right there. PdhS2, we would predict the opposite, and I'm not going to show the pull-up, but it looks quite different. And you can see that the green foci are scattered throughout the surface of the biofilm because the bacteria are dividing. So there are new poles all throughout the biofilm. So that does lead us to this particular model, and the model being dictated and controlled by localization of proteins. A couple questions that come up here, and this won't take too long, is that, for one, we have this response regulator X. And that is something that some data that I didn't go over led us to hypothesize that there's at least one other response regulator that is interacting with this pathway and is controlled by the PdhS kinases. So we have kinases interacting with DivK. We believe they interact with some other response regulator X. We have two candidates. One of these response regulators is studied by one of the grad students in the lab, and the other we're not studying at all, but we are following up on at least one of them. The other question is, we're producing this small molecule cyclic di-GMP inside the bacterial cell. Levels of cyclic di-GMP, high levels, promote biofilm formation and inhibit motility in agro and in many other cells. And so a natural question is, oh, actually I can pull it up. The natural question is, well, how can we determine how are these cyclic di-GMP levels cleared? I mean, if it's producing this message, you got to get rid of the message at some point. You can't just have the signal always around. And so one way that we do that is we did that. There we go. Using swim motility suppressor, where we have a mutant that can't swim very well, the PdhS2 one, for example. We get active flares of motility. We isolate those flares and determine what's mutated, what's changed, what's interacting with this pathway genetically. I'm going to skip through this because that's old data, but focusing on the cyclic di-GMP, we have used that swim suppressor assay to identify a couple candidates, one of which has both a GGDEF motif and an EAL motif. So synthesis of cyclic di-GMP, this second messenger inside bacterial cells, it takes two molecules of GTP and then a diguanylate cyclase with this GGDEF motif, puts these together into circular form. That's cyclic di-GMP. That is the messenger. This can then be degraded by certain cyclic di-GMP specific phosphodiesterases. And in one of my classes, we actually discovered a really promising hit called ATU3207. And ATU3207 is a cyclic di-GMP specific phosphodiesterase. We believe, and all of our data support, that this protein ATU3207 degrades cyclic di-GMP concentration. What's more, if we overexpress it, you reduce biofilm formation. That's cool. If you delete it, it increases biofilm formation. Nice there. It is polarly localized. I'm showing you some data here where you can see polar localization of ATU3207 and a couple of those there. It is expressed in a CTRA-dependent fashion, so it is cell cycle expressed. And this is just saying that we found it in a class. So how are cyclic di-GMP levels cleared? Well, we think ATU3207 is one way it's cleared. All right, I think that comes back to that model that we did. This is future work of what we'll be doing. We want to continue to validate the activity and targets of these proteins and make sure we know how all these pathway components fit together. We have no real idea how localization happens. We have some ideas, but not some data. We want to look at gene expression within the biofilms and while interacting with the host, much like that one study that was earlier that I was super excited about. We want to measure fitness contribution in the rhizosphere, which means we want to do greenhouse work. So anybody who can get us a greenhouse, hint, hint, that would be useful. And I'm actually really interested in focusing on citrus greening in the future, which is a disease of citrus. Imagine that. It's caused by an alpha proteobacterium called Liberibacter, different species. It's a huge issue, okay? It is literally decimating, i.e. reducing to a 10th, our crops of citrus fruits. And it's really hard to deal with. It has the CTRA pathway. So maybe we could translate our work to that organism. That would be great. And just everything I showed you so far was genetics and phenotypes. But I just want to, put up my biochem bona fides and say, look, we're purifying proteins. And we want to actually mix our purified proteins and make sure if I say something phosphorylates something, we want to say, oh yeah, look, we phosphorylated it. So here's just showing an example of purification of CTRA and purification of DivK. So we're getting there. We're not there yet, but we're getting there. And that I will definitely stop there. Maybe if I can get, go, there we go. So that's our pathway, controls these phenotypes. We could talk about all other things. I don't have a couple more hours. This stuff that's going on here, the VTLR pathway fits in right here with oxidative stress. So if you're working on VTLR or interested in it, that's kind of where it fits in the whole thing. Here are the peeps, the team, and our funding. And I'll stop there because I went way over my time. Too bad you only get to do this once every now and then. So captive audience. Okay, I think we have time for a question.
We have until we have 10 more minutes.
Okay, I'm happy to take questions from any students.
Students first, and then if you could repeat the question
Yep.
into the microphone.
But non-students, okay.
And you have to say microphone that it's on Zoom.
Yeah, if you're on Zoom, you can write to Greg and we will chatify your question. I know I went through a ton of stuff. I kept it higher, yeah.
So I was wondering that you can study CTRA, but can you also study like a substance that we can treat the plant with that would disable the binding of T cells that we could protect it from danger?
Yeah, so the question is, can we, instead of looking at maybe the genetic pathway, could we do something that could abrogate or completely abolish binding of the bacterium with the plant tissue? So like treat the plant surface with something that prevents UPP deployment or stickiness. We could. Yes, that's the quick answer. Yes, for sure. And there are a number of researchers studying the specific physical properties of the surfaces that allow for attachment and allow for maybe surface treatment. So material scientists, that's something that they think about all the time. And we have material scientists in the building.
And just one point, you say that the transportation efficiency is like 4%. Does that mean that the bacteria is not just for the plant?
So the question is about the transformation efficiency numbers that I put on earlier in the slides. And if they're so low, does that mean that the bacterium is not dangerous for the plant, or what do those really mean? So those are in experimental conditions. I don't know that anyone has determined what's the actual efficiency out in the real world. My guess is it's actually pretty low, but that's okay because there's a lot of agro out there. And it can live in the absence of the plant host. That's a good question. Yeah, Claude.
I have a question. So, with regards to the RNA-seq data.
Yep.
So you say bacteria will attach to the plant. They will upregulate everything to transform the plant so that the plant cells divide, which provides food to the bacteria, a great environment for the bacteria. But the bacteria down-regulates everything that's needed for division. So what's the point? The point is not to make more bacteria. Because usually when you perform these efforts, it's to promote reproduction.
Right. So the question is, with the RNA-seq data from this 2018 paper, it's this conundrum of division appears to be down-regulated at the point of interaction with the plant tissue. But the organism is making the plant make resources so that it can presumably grow and divide. And that's a great question. I think that what they've got, they don't have a synchronized population of cells. So at the surface, when they're interacting with the plant tissue, they've got, like you can imagine within a biofilm, the ones stuck at the bottom might not be dividing. The ones a little further up might still be active and dividing. So I think there's heterogeneity that is not able to be seen in that RNA-seq data set. So that's my hand waving at that answer for now, but it's a good question. Yeah.
I have a question from Energy. Says, how old from an evolutionary perspective is this bacterial behavior that attacks the GFOs via the UV biofilm? Well, wondering mostly if it is something that evolved after plants were a thing, or if there were some evolutionary agents.
Can you repeat the first part again?
How old is the evolutionary perspective of this bacterial behavior?
So how old is the behavior of bacteria Attaching. I don't know. That's a good question. I'm trying to think of how to address that because so many bacteria interact with so many different host cells. And I would say, yeah, I don't know. That's an interesting question because like I'm thinking I can answer more specifically towards CTRA because CTRA activities can help the bacterium escape phagocytosis by amoeba, for example. And presumably there were predatory amoeba around pretty early on once you had a eukaryotic cell. I don't know that I have a good answer. I don't have an answer. Sorry. Go
How
ahead.
prevalent is that UPP in other?
The UPP, the unit. So the question is, how prevalent is the unipolar polysaccharide in other bacteria? It is almost I wouldn't say universally. It's well conserved among the Rhizobiales, and the Colobacteriales. So the ones that are closely related to agro, but it's not so well conserved further afield. But Brucella has the genes for it, so you can go a little bit out. Yeah.
So when we think about the great connection here, way at the beginning, maybe the first slide, you said something about how the agro would introduce a gene for the plant hormone.
Mm-hmm.
How the agro get?
That is okay, anyone who's taken microbiology, how did agro get that? Two possible mechanisms, three really: transformation, transduction, or conjugation. So horizontal gene transfer, presumably. Sorry, online, the question was, how did agro acquire the plant hormone genes? And so, you know, I haven't delved too much into how closely related the genes that are causing synthesis of those hormones are to the plant genes that synthesize those hormones. That's an interesting question. I'm sure that is known. I don't know that. That's an interesting, that I'll look into that.
So
Go.
the question, kind of relates to in some sense what Paul was asking, but I guess I'm kind of interested in what about the daughter cells that are budding off, right? Because if you've got some like pre-living stuff and stuff that's reversibly down, there's a decision point that you're making here. This daughter cell can be, but I probably want to shoot right back to it. And things just like shift, and I'm like, okay, that right? So
Yeah. So the question is what is known about the fate of daughter cells that are presumably at the surface and dispersing from or budding off from a microcolony or a biofilm attached to the host? Is that good phrasing? Not much is known. Cartoons are known. Speculation is known. But exactly the way you framed it is very well framed. Is certainly the daughter cell can sense the environment. And so based upon what it's sensing and what it's interacting with physically, it could be trapped near the surface and then become part of the biofilm again. Or if conditions are favorable to disperse, then it could disperse. But I don't, it's an interesting question, particularly in the rhizosphere. Nobody studied that.
and I think especially, you know, like what are the triggers right in the environment that they're really, to make the decision?
Yeah, I mean, the secreted molecules from the plant, like the acetylsyringone and other phenolics, those are the known ones. Yeah. Okay, two more, go. Who's first?
How do we know how the PdhS kinase moves from one pole to the other? Because it's rapid and directional, but there's no vesicular movement and there's no cytoskeleton or actin filaments. How does it move?
Yeah, so we don't know. So the question is, how do the PdhS kinases that move, like PdhS2, how do they move from A to B? So I've done sleight of hand. I told you that they move. We don't know that they move. It could be that it is degraded from one pole and then resynthesized and inserted at the new pole. So we don't know that yet. But if you look really closely, you actually see as the one pole focus is dispersing, the periplasm turns green. Or not the periplasm, the membrane turns green. So I actually do think it's actually being redistributed in the membrane. It is membrane bound and has a transmembrane domain. So I think it is actually moving in the membrane. I don't know that. That's my hypothesis. We haven't tested that. So we don't know. It's a great question. Yeah. So what happens to the bacteria if a phage comes and and infects it, well, then the bacterium dies. Pretty much, if it's a lytic phage. And there are bacteriophage that are agro-specific. There is no transducing agrophage. So if you ever find a phage that can transduce into agro, the community would love you. But yeah, the typical infection process that would be caused by a bacteriophage infecting that bacterium host, it would most likely become lysogenic or ultimately become lytic and then kill the host.
We are out of time. Thank you so much for being here. Let's give it a hand.
Okay, we're